Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. In this video, we will show you what is enzyme assay and how we are conducting enzyme assay acetylcholine esterase before purification in the lab session. Before we going further into the process, let me have an introduction. What enzyme we are using in this enzyme assay is we are using acetylcholine esterase, which in common called ACHE. This ACHE can be found in the catfish brain. Next, the purpose of this enzyme assay is to observe the enzyme activity that happen at different concentration of ATC in the lab session. Now, principle of enzyme assay. As I said that in this enzyme assay, we are using acetylcholine esterase that have been extracted from the catfish brain. As you see, in the procedure, at the end of the addition of reagent, there is formation of yellow colored product in the cuvette. So why it is yellow colored product? It is because ACHE will break down the acetylcholine ATC into thiacholine and also acetate. This thiacholine will later actively react with the DNB reagent to form a yellow colored product. This yellow colored product will measure the intensity by the spectrophotometer. So in some high color intensity will indicate the high enzyme activity.
I will briefly explain the materials and reagent used in the enzyme assay. First, let's look at the reagents. The first reagent is tris buffer and is used to maintain the optimum pH of ACHE. The second reagent is the crude enzyme extract, which is obtained from the catfish brain and will serve as the source of our ACHE in this experiment. Next, we have ATC. ATC is a substrate for the ACHE. During the reaction, ACHE will hydrolyze ATC to produce thiacholine and acetate. We also have DTNB, which is also known as the Elmer's reagent. DTNB reacts with thiacholine and will produce during the reaction and will form a yellow color product known as TNB. We also use deionized water to prevent the reagent solutions and prevent interference from impurities. Now, as for the materials, a spectrophotometer is used to measure the absorbance of the yellow color product, and this absorbance value is then used to calculate the enzyme activity. Micropipettes are also used to accurately transfer small volumes of reagents and samples. Beakers and conical flasks are used for preparing and mixing the solution. And finally, we have cubates, which is used to hold the reaction mixture during the absorbance measurement.
Moving on to the procedure. For this enzyme assay, we perform the reaction directly inside the cuvettes. We set up a total of 4 cuvettes, which is 3 replicates label 1, 2, and 3, and 1 negative control label C. First, we pipette 40 microliters of our crude enzyme extract straight into the 3 replicate cuvettes. Next, we pipette 900 microliters of 0.02 molar tris buffer into all 3 replicates and 940 microliters into the negative control cuvette. After that, we add 50 microliters of 0.33 millimolar DNTP solution into all 4 cuvettes to serve as our color producing indicator. Now, to initiate the enzymatic reaction, we add 10 microliters of ATC substrate directly into the cuvette. Immediately after adding the substrate, we mix the solution quickly and thoroughly using a micropipette tip and place the cuvette straight into the spectrophotometer holder. We take our very first absorbance reading right away at 0 seconds at a wavelength of 405 nanometers. Because this is a continuous kinetic assay, we monitor the real-time progress of the reaction by recording the absorbance reading every 10 seconds of a full minute. We repeat this exit direct cuvette pipetting and measurement step individually for replicates 1, 2, and 3. Running this experiment in triplicate is highly crucial to ensure the precision and reliability of our kinetic data. Since we are dealing with a very small volume of substrate, which is only 10 microliters, doing this helps us to minimize random pipetting errors.
My name is Akhilesh Chandrasekhar. My number metric is BC250873 and my part in this presentation is results and calculations. Table 5.3 and figure 5.3 shows the kinetic behavior of acetylcholinesterase ACHE at different ATC concentration. The enzyme activity increased with the increasing substrate concentration and reached the highest reaction velocity of 0.0814 micromole per min at 0.8 mm ATC. However, the activity decreased at 1.5 mm and 3.0 mm ATC, suggesting substrate inhibition due to excess substrate. The kinetic analysis gave a Vmax value of 0.0677 micromole per min and a Km value of 0.0395 mm. The low Km value indicates that ACHE has a high affinity for ATC because only a small amount of substrate is needed to achieve half of the maximum reaction rate. The low sum of errors 0.00749 also shows that the regression model fitted the experimental data well. Overall, the results indicate that ACHE exhibits optimum activity at 0.8 mm ATC and has a high affinity towards its substrate. Thank you.
Next, I will talk about the factors affecting our enzyme assay when working with the crude brain extract from the catfish. When working with a crude brain extract before purification, we are dealing with a complex biological mixture that introduces severe interference into our kinetic data. First, we face proteolytic degradation. The moment we disrupt the catfish brain tissue, cellular compartmentalization is lost, releasing active endogenous proteases. These proteases will rapidly degrade our target acetylcholinesterase, leading to a false underestimation of its true activity. Second, we must contend with high lipid turbidity. Because brain tissue is highly rich in structural lipids and myelin, the raw homogenate forms a cloudy emulsion. Since the standard assay relies on a clear light pulse to measure absorbance, this cloudiness scatters the light and skews our spectrophotometric readings. Third, there is significant non-specific background interference. The colorimetric reagents used in this assay react directly with thiol groups. Since crude brain tissue contains high concentration of non-target proteins and free antioxidants like glutathione, it triggers a massive background reaction that masks the true initial velocity of our enzyme. Moving on to the essential precautions, we must implement 4 strict operational safeguards to neutralize these threats and ensure accurate data. Number 1 is strict temperature control. We must maintain the catfish brain tissue homogenization tubes and buffers between 0 and 4 degrees Celsius at all times. Keeping everything on ice suppresses the activity of those destructive native proteases. Number 2 is targeted buffer chemistry. Our extraction buffer must be reinforced with a robust protease inhibitor cocktail to protect the enzyme structure, alongside a strong chemical buffer to stabilize the optimal pH. Number 3 is complete clarification. High-speed centrifugation in a refrigerated unit is mandatory to cleanly separate and pellet down the dense lipid and cellular fragments, leaving us with a clearer working sample. Finally, number 4 is the use of mandatory sample blank. Because the crude catfish brain extract has its own native turbidity and color, a standard water blank is insufficient. We must run a dedicated sample blank containing the extract and buffer, but no substrate. This allows us to measure and mathematically subtract the extract native background absorbent from our final kinetic calculation.
In conclusion, this experiment successfully determined the activity of acetylcholinesterase or ACHE from catfish brain extract before purification. Using the assay method, enzyme activity was measured at different concentration of acetylcholine or ATC. The results showed that enzyme activity increased with increasing substrate concentration and reached its highest value of 0.0814 micromole per minute at 0.8 millimolar ATC. Beyond this concentration, enzyme activity decreased, suggesting possible substrate inhibition. The Michaelis-Menten analysis produced a Vmax of 0.0677 micromole per minute and a Km of 0.0395 millimolar, indicating that ACHE has a high affinity for the substrate. Overall, the experiment successfully achieved its objective of evaluating the activity and kinetic behavior of ACHE before purification and provided a better understanding of enzyme kinetics. That's all from us. Thank you.